These are the lecture notes for Chapter 14 on DNA structure and function. A little bit of history. In 1869, Friedrich Miescher isolated phosphate-rich acidic compounds from the nuclei of leukocytes, and leukocytes are white blood cells. They're a type of white blood cell. In pus from hospital bandages, and he called this material nuclein since he was able to isolate it from the nuclei. But we know today that what he isolated was DNA, or deoxyribonucleic acid. It was many years until we knew that that's what he had isolated. Now, some of the first experiments with DNA were done by accident, and they were also done. The purpose that we use them now is to prove that DNA is the genetic material. We knew that it was either DNA or protein, but we didn't know which one. So anyway, this is a experiment that proved something called transformation. Okay, so Frederick Griffith used 2 strains of Streptococcus pneumonia, which is a bacteria that is deadly to mice, or that one strain is deadly. And he injected mice with heat-killed virulent S strain. And so because the virulent S strain is the deadly strain, so because they were heat killed, he exposed them to really high heat, the mice lived. But then he also injected mice with heat-killed S strain that had been mixed in the same test tube with live non-virulent R strain, and that killed the mouse. So by itself, the non-virulent R strain doesn't kill the mouse. By itself, the heat-killed S strain doesn't kill the mouse. But when mixed together, something happens to the S strain DNA, and it is actually taken up by the live R strain, and so the live R strain becomes virulent. And this process is called transformation. And then we needed to know what was the transforming, what was the chemical that was responsible for transformation. And there are many experiments that were done that together proved that DNA is the actual chemical that's responsible for transformation. It's actually the DNA from the S strain that is infecting the R strain and making it virulent or deadly. The scientist that showed that the transforming principle was most likely a nucleic acid were Oswald Avery, Colin MacLeod, and Macklin McCarty, and this was in 1944. They recovered cell extracts of the S strain from the dead mice. They used enzymes to degrade macromolecules, and they mixed these extracts with the R strain and injected the mice, and they got the same results. That it killed the mice. Now, another experiment that proved it was DNA and not protein. This was an experiment specifically to prove that the genetic material is DNA, not protein. And the way that Hershey and Chase went about this is, I'm showing you this picture first instead of the notes, because this is what a virus looks like. Okay, I was just looking to see if we had a bigger picture, but this is what a virus looks like. A typical virus. Viruses have different shapes and that kind of thing, but basically the black capsule on the outside that's kind of shaped like a hexagon, is made of protein. So that capsid is protein. And inside the green squiggly line is the DNA. So basically, a virus contains protein and it contains DNA. So it's one of those that is responsible for tricking the host cell into reproducing new viruses. So Hershey and Chase did an experiment to prove, to determine whether or not it was DNA or protein. Okay, so they used bacteriophages, and bacteriophages are viruses that infect bacteria. Viruses that infect bacteria. They're called bacteriophages. So we think about bacteria as causing us to get sick, but a bacteriophage is a virus that causes bacteria to get sick. So these bacteriophages were used, and basically they were infected with um, these bacteria were infected with bacteriophages that had been radioactively labeled with radioactive sulfur or radioactive phosphate. Now, the radioactive sulfur would be found in protein. So anywhere that you could trace that radioactivity, you would know that that chemical was a protein. And the radioactive phosphorus is found in DNA. So anywhere that you could trace the radioactive phosphorus, you would know that it's DNA. So they determined what had entered the bacterial cells, and the only thing that had actually entered the cells was the radioactive phosphorus. So that indicated that DNA is the genetic material, because it would be the chemical that enters the cells that actually causes the host cell to reproduce new viruses. Because that's what viruses do. Viruses trick the host cell into producing more viruses. So if you look at the experiment now, experiment 1 is when they used radioactive phosphorus, radioactively labeled phosphorus, um, which can trace the DNA, and they found out that when they inserted the viruses or mixed the viruses in a test tube with the bacteria, and then they used a blender so that the heavier particles fell to the bottom. The heavier particles would be the cells. Then the bacterial cells could be isolated, and they found that the bacterial cells contain the radioactive phosphorus. And when they labeled the radioactive protein coat sulfur, they did not find that in the bacterial cells. So it was proven then by this experiment that DNA was the genetic material. DNA is the chemical that contains the code for all of our traits. Now, Erwin Chargaff, discovered that when you analyze DNA, that there are 4 types of bases. We call them bases. There are 4 bases in each nucleotide. There are 4 types of nucleotides that can be found in DNA. So DNA is a combination of A's and C's and G's and T's, just billions of these 4 chemicals: adenine, thymine, cytosine, and guanine. And what Chargaff discovered was he analyzed DNA, and he found out that when the percentage of adenine, say, was 20%, then the percentage of thymine would also be 20%. And in that case, since 20 and 20 is 40, then cytosine and guanine together would add up to 60, because percentages are out of 100. So the percent of cytosine would be 30, and the percent of guanine would be 30. So in other words, he found out that the percent of adenine was always equal to the percentage of thymine, and the percentage of cytosine was always equal to the percentage of guanine. So you can always, if you know the percentage of one of the nucleotides, then you can figure out the percentage of the rest. But what this told us is it told us how it gave us a clue as to how DNA is arranged in its double helix shape. Now, one thing we know is that DNA is a nucleic acid, okay? And nucleic acids have building blocks called nucleotides, okay? So a nucleotide is made up of a sugar. In DNA, that sugar is deoxyribose, and in RNA, that sugar is ribose. And a phosphate group, I can't underline it, I'll put it over, a phosphate group, and then a nitrogenous base, we'll just say a base, and that base can either be A, adenine, T, thymine, C, cytosine, or G, guanine. And in RNA, instead of thymine, it will contain uracil. So RNA could contain adenine, uracil, cytosine, or guanine. Okay, whereas DNA would be adenine, thymine, cytosine, or guanine. Now, we call 3 of these bases pyrimidines because they only have a single ring structure. They're shaped like hexagons. Cytosine, thymine, and uracil are pyrimidines. And then we call the other 2 bases, adenine and guanine, purines because they have a 2-ring structure. This is an X-ray image that was taken by Rosalind Franklin. And this image helped James Watson and Francis Crick determine the structure of the DNA molecule in 1953, for which they won the Nobel Prize. Sometimes I'll get students to ask why Rosalind Franklin did not also win the Nobel Prize, and she was not included because she had actually passed away from cancer. They think because she did work around so much radiation doing her research. But she passed away from cancer before the Nobel Prize was awarded, so she could not be awarded the Nobel Peace Prize because that's one of the rules. Some people think it's because she was a woman, but and that could also have been the case, but it wasn't. You know, it's just she just happened to be she happened to die, so she was not awarded the Nobel Peace Prize. Now, what Watson and Crick discovered, the DNA structure, is that DNA is a double helix. It's a polymer, meaning it's a very, very long, very large molecule. And it's made up of repeated nucleotides that are bound by phosphodiester bonds, and they are anti-parallel to each other. One strand runs from the 5' to the 3' direction, and then the other strand runs from the 3' to the 5' direction. So anti-parallel. They're parallel, but they run in opposite directions. And the 5' and the 3' are just labels for the carbons in the rings. So that would be the 3' stands for the 3rd carbon, and the 5' stands for the 5th carbon. It's just a way to determine position, basically. So what you also see is that every time there is a thymine, it's going to bond with an adenine in the double helix. And every time there's a guanine, it's going to bond with a cytosine. And that keeps the steps of the ladder, or the rungs of the ladder, having a total of 3 rings, so that the rungs of the ladder are always the same length. So A's always pair with T's, and C's always pair with G's. And of course, in the case of